Good morning. I was just checking he was telling the truth. Okay, today's talk is the aim is to start a conversation. It's to start a conversation. It is my experience. As I said, I have a lot of experience in this, and I've seen a lot of false hope in the varroa story, where people have promised things which have never come true. So when Melissa ruined my life 8 years ago, or 6 years ago, I thought she was correct, but I needed to check. So I was very fortunate to be in a position to travel and to visit places. And so the last 4, 5, 6 years, me and my very small team have been very busy trying to see if what Melissa's idea was worked. I think we're very confident it does. And now we'll try and pass why I think it works. And the idea is to start a conversation. We've got a conversation started in the UK and other countries, and so now it's your turn. Okay, so varroa resistance. And I'd like to say this research, these last few years, it's all funded by beekeepers, UK beekeepers. And so this is beekeeping science for beekeepers, funded by beekeepers. So we heard yesterday there's a problem with varroa. Hands up those in the audience that are old enough to have kept varroa kept bees before varroa arrived. 1, 2, 3, 4, 5, 6. A very small number of you. And if you can remember, these people can remember, when varroa first arrived, they did not want to treat. They wanted a solution. They wanted a silver bullet. And it didn't happen. A lot of people who didn't treat lost their colonies. And it took a long time and a lot of persuasion for people to start treating. Now, it's evidenced by everybody in the room that their prediction at the time that their beekeeping would collapse, there would be no beekeeping anymore, is wrong. There are many beekeepers. But the majority of people here treat because that is how you keep colonies now. But you're actually in the minority in some way, and I'll try and explain why. The problem I have now is trying to convince you that actually now's the time not to treat or to start reducing treatments. And that is really proving difficult. Okay, so the solution. So now we have the solution, believe it or not, and we also know the problem that's causing things. And I'm sorry to say, it's you, it's the beekeeper. I must say that if it wasn't for you, there probably would possibly not be many bees left, or they could be doing extremely well. We just don't know what the situation would have been in Denmark. We know, I think it was Slovenia or one of the small countries decided not to treat, and they lost the vast majority of their colonies, and eventually they had to start treating. So the solution is for all beekeepers and for any race of honeybee. So you will be very happy, because that means your buckfast bees will be able to come resistant. You won't have to buy resistant Carnica queens in from the Germans or the Norwegians or all the other countries that you appear to hate. So this is something that's often lost in beekeeping, is that Apis mellifera is a single species, as we were talking about all the variants in the human race and the variants in the virus, and you've got the variants in Apis mellifera. But they are just one species, and all the different bees are able to do this. You can do it in any environment. What we often forget, that inside a colony, a bee colony, is that the conditions in Alberto, in -20, or in the tropical Amazon, it's almost the same inside the colony. So the mites are experiencing a very similar environment. So this is again very fortunate, and it's in any type of hive. I have sat through countless lectures over the years about how this hive and that hive's better, and this has got big advantages, and etc., etc. So you can all keep using your wild and wacky different types of hives and keep resistant bees in these strange boxes. And you can do it anywhere in the world, which is, again, a really great thing. And this is why it's called a silver bullet. It's one solution for everybody. And all you need is this. So if you've got this, if you've got some bees, you're halfway there. And hopefully you've all got bees, otherwise you wouldn't be here. Although we do have beekeepers without bees. And you need something else, and this something else is knowledge. And today we will start to help provide that knowledge, and you can start a discussion, start looking at the information and make your own personal choice about this. So first of all, varroa resistance in Apis mellifera has been around since the 1980s. I was studying it in the 1990s. The whole of the area where Africanized bees were, they're completely resistant to varroa. Nobody treats. There's no need to treat. And this is a huge area of the world. And there is a huge amount of organic honey exported from these countries because they don't have to put chemicals into it. And it's a godsend for the honey markets, places like Mexico and Brazil, where it's a big trade. Sub-Saharan Africa, right down to South Africa. Now we've confirmed it in South Africa, but in all of Sub-Saharan Africa, people don't treat. And people can't treat. They don't have a choice. The whole of the Caribbean, in fact. So where there is poor people in the world who can't afford to treat, they've had to rely on their honeybees becoming resistant, with a couple of exceptions I'll talk about. So by the sort of 2010s, it was very, very widespread and increasing. So there are pockets of resistant bees which have not been treated for at least 6 and often more than 10 years in America, in Europe, in the UK, which is now separate from Europe, don't take me there, and Italy. And the more you ask, the more you find. And we know that in temperate climates, in European countries and America, it's hidden. There's a hidden group of beekeepers. So we did a survey. I got one of my third-year students to do a survey, Alex Valentine. She sent out via email to associations, and they responded, and we got a response from about 3,000 beekeepers in the UK, from all over the UK. And we were asking about their treatment habits. Now, this wasn't perfectly designed survey, it was just the sort of first see. But what we got was sort of partly what we expected. So about 3/4, about 75%, actually 72 in this, but 75% of British beekeepers, because it was all over the UK, were treating. And they were treating once or twice a year, mainly with oxalic acid, some with formic, but mainly chemical treatments. But there was a group of 1 to 5 year olds, so that they hadn't treated between 1 and 5 years. Now, some of these, their colonies would die, some of them would need treating. But this is very promising because it means that 25% of the people in England are at least trying to go down a sort of treatment-free route. But what's really important is this little group of the 6 to 10 years never treated and the 10-year plus never treated. And this is 6%. Now that equates to about 1,800 beekeepers in the UK. We know personally of maybe 20. So there is a lot of hidden people out there, and it makes sense. Everywhere I go in the UK to give a talk, there's always one person comes up and tells me secretly that they have bees they've not treated for a long time. And then we have a small group that have never administered treatment. These are part of the natural beekeeping movement who just provide homes, and some of these colonies will die and some won't. So we looked at the distribution. This is the very busy map of the UK, but light blue gives an idea of the proportion of the people in that association that are not treating or trying to go down a non-treatment group. And they're everywhere. Almost every association has somebody that is trying this. There are only 2 associations, I think 3 associations, where the majority is actually treatment-free. And I'll talk about this in a minute. There's a group in North Wales here in the Llyn Peninsula. There's the Swindon Conservation Group down here, and then there's this Westmanham group I'll also talk about down here. So this is good. So there's, for the UK situation, there you should be able to expand this on a very local scale. And I suspect, maybe not as much, but in Denmark you will have something similar. Might be a smaller number, but you will have a small number of people that have not treated for many years, that have stayed silent. And for good reason. They stand up a bee meeting and say they've never treated for 10 years, they will get shouted down. You're a bad beekeeper, you're going to create mites, etc., etc., so they don't talk. This is one example. So this is a very well-known group of beekeepers. They now are 104 beekeepers. They total about 500 colonies. This is an area in North Wales, quite a large area in North Wales. And they started by collecting feral colonies, wild living colonies from trees. And then they passed them colonies on to other people, and basically by a friendship network, and they gave bees to other people, and they were resistant. And nobody treats in this entire area except one person. There's always one. One person still treats, but that's his prerogative to do that. And I have a nice story I've just got from one of the beekeepers there. He said, I think 5, 6 years ago, he started beekeeping, moved into the area. He was mentored. He was given a couple of nukes. Then they survived. He split them when they became colonies, and he split them colonies. He got a couple more, and now I think he has 15 colonies. And he says at the end, I wouldn't even know how to treat for varroa, because he's never had to, because he's been very lucky to move into an area where they've already got resistant stock. So this is possibly a glimpse into the future. So the mechanism, let's see how this works. So it's important that we understand, and we already do from yesterday, the role of the virus, the mite, the honeybee, the beekeeper, and a thing called hygienic behavior, but not as you know it. You're thinking as hygienic behavior maybe with freeze brood kill. It's not the case. It's something similar. It's a variation. Hygienic behavior is a general behavior. Okay, so there's this brand new paper that my student has just finished and just got published. So it's the first time I've shown this slide. And what she's done, she's gone through all the literature and she's had a look at the resistant traits that are in Apis mellifera. She's looked at the ones, compared them to treated Apis mellifera, and actually to that in Apis cerana as well, which is naturally resistant. And we know yesterday from Melissa that recapping seems to be a key trait here, where it is present in Apis mellifera, but in resistant Apis mellifera, it is much higher. Apis cerana, we have no idea. But what's surprising from this is grooming. There is a lot of people done work on grooming. And the difference between Apis cerana and Apis mellifera treated and resistant is not that large. And in fact, a lot of the data, the entire grooming data on Apis cerana is based on 3 studies. And they're all flawed in some way. So it's quite interesting. And then brood removal is important, and then mite fertility is also important. So first of all, I'll cover all them different aspects. So first of all, grooming, and I love this story. So there's a lot of people who are still, and have done in the past, looked at these dimples on the mite and used them as direct evidence that this is where the mite, the bees are biting the mite, and they've used these for selection. This is a very bee-centric, beekeeper-centric view of things, where they think the bees are really good at this. And it turns out, an acarologist pointed out that this actually is a birth defect. It has nothing to do with grooming. And where these dimples are, they're very symmetrical, or they can be. They're either on one side, as in this one, or they're on both sides. And it's exactly where the muscles, the very large leg muscles for varroa are. And if there's not enough hemolymph in the varroa mite, there are dimples caused. You can find these in in cells, sealed cells of pupae. And so the mite never had opportunity to be exposed to bees. And the other one is this, is that I had a student many years ago, and you can see the mites here very, very clearly. But you're a visual animal. Bees are a chemical animal. Their world is chemistry. And this is what the chemistry is. It's like a chemical barcode of a honeybee at the top and of a varroa mite on the bottom. They are incredibly similar. And in fact, when we look at them in great detail, they're identical. So chemically, the varroa mite inside the cell, inside the colony, which is dark, the bees cannot see them. They can feel them to start with until they get used to them. But as I say, these mites are highly adapted. And this explains why grooming just doesn't work. So I'm not sure this will work. When an invading bee comes into the colony, which is a brown one, might be carrying a mite. Oh, there it is. Very hard to see because it's blended to that particular odor of the colony which that bee came in, or it could be a newly emerged bee because they smell slightly different than the adults in the colony. Then the mite moves onto a new bee. Now, it can be seen. It is chemically somewhat different, but it has suckers. It can live on the underside of the body, which is very difficult to remove. It has lots of adaptions, and it only needs to use these adaptions for 3 hours until it blends in with that new colony. As I mentioned, this is a highly specialized mite. And this she used to call the ghost in the hive because the bees cannot see them. And this is why grooming behaviours are often flawed. They do it over a very short period of time, and I'll stop there. So I put these in for somebody who asked a question yesterday. What would happen if there was no deformed wing virus? I did touch on it very briefly, so I'm just going to go through this very quickly. There is a small island where that star is off the Brazilian coast called Fernando Noronha. It is extremely hard to get to, and it's got European honeybees there. They were taken there to set up a queen rearing station. That failed. They had varroa mites there, but they persisted, and they've persisted for 25 years. The mites have been at the same level. There is completely no losses. The beekeeper, he doesn't even know what varroa is. And when we checked the bees, deformed wing virus was absent. This is very rare. And mite resistance has appeared. Now, at that time, we didn't know about Melissa's breakthrough, so we've no idea what actually is going on there, the actual resistant mechanism. But we do know that their mite reproductive rate, which, if you remember, over 1 is bad, and the mites are going to expand. Below 1, and the mites are basically not a problem. 0.5 means they're just about hanging on. And there's a huge a number of infertile mites at this place. So they can have actually quite high infestation rates, but because they're not transmitting the virus, they're not actually doing any damage. Now, there's also been two other cases. Deformed wing virus is also absent on the Solomon Islands, and they don't have a problem with varroa, and actually on Papua New Guinea in the Highlands. And again, that's slightly complicated. It's a different varroa, but it's the same system. So let's quickly go on to the mechanism. So this is the key. This is still a work in progress, as in the precise details have not been worked out. There's been a lot of work done by Marla Spivak, and I'm using that approach. But it's all basically semantics. It's just that the detail, but we know how this thing works. So you have bees which are sensitive to smells, different smells in colony, be it a dead bee, the AFB, chalk brood, or wax moth. And they will investigate that cell. The bee has to learn the odor. So they've had to learn a new odor associated with varroa or varroa-infested cells. And what these specialized bees do, Marla Spivak showed very nicely, that for a short period of their life, a couple of days, they become very sensitive to smells. And these become very specialized, and it's to do with the changes in their brain chemistry. So these bees are really good at detecting differences, and they have learned that this odor is associated with an infested cell. And so they open it to get a better smell, because these compounds have got to go through wax, which tend to absorb chemical smells. They make a hole, but also they make a hole in surrounding cells, which again we heard from Melissa yesterday. These non-infested cells can be recapped, and I'll explain. Now we've got cells which are open in the colony. Now there's two choices. So the specialized bee now moves off. Now we've got other bees that will see these holes and just recap them. They'll go, oh, there's a hole, larva's not dead, pupa's not dead, no problems. Okay, don't know what's going on here, let's recap it. And they recap it, even if it's got mites in it. While there's others that will think, aha, there's something wrong with this cell that's been opened, let's investigate, aha, it's infested, and they cannibalize it. They actually eat the pupae down. And as they're cannibalizing it, the mother mite, unfortunately for you, escapes, because she's highly adapted, but her offspring are not, and she loses all her offspring, and this is key to this. They clean out that cell, and sometimes, well, quite often in resistant colonies, you'll see this. You'll see half-eaten pupae, and often you'll see mites, evidence of mites in there, or the mite offsprings. And you can actually pick these things up off the varroa floor, and I'll show you pictures later. And so you've got this removal. And this doesn't happen once. This happens repeatedly. I was talking to Ralph Butler over in Germany, and he says they were videotaping this behavior, and they had one cell that was uncapped and recapped 17 times during the development of the bee. Okay, this is a vast extreme, but it does happen repeatedly. So it may make a mistake once, but it keeps on going, because then the sensitive bee comes along and goes, hey, what's going on here? This needs to be opened. They reopen the cell, and you just hope a recapper doesn't come rather than a remover. And you have no control over this, because we don't know which bees are recappers and which bees are good at doing sensitive. But one of the consequences of this is you often see this associated, not always, but more frequently, with resistant colonies. You see these patches of bull brood. Now, you'll all have seen these, because these became more common after varroa arrived, because all bees can do this. And what this is, and what Marla thinks this is what happens is sometimes there's just not enough recappers, and so these get opened and they get left. But if you come back in 2 or 3 days' time, you'll find that the vast majority of these cells haven't disappeared. They've just been recapped over. And they're usually of the similar age. They usually have a sort of a pinkish or a purplish eye. Never usually younger, and they're never usually older. But there's always some beekeeper will tell me differently, that they did once see a bee that was just about to emerge, that, you know, was opened. But generally, they tend to be of this pupal age. Due to some fantastic work done by Fanny Mondet, a big French group, they actually know what the odor cues are that are produced from the infested cells. They are unique compounds to honeybee colonies. So they're not found in honeybees colonies normally. And this is this group of ketones and acetates. So we know what they're doing. She showed very nicely that all bees could detect these compounds, but only resistant bees knew what they meant to actually change their behavior. And this is part of, if you want to become resistant, you've got to give bees time to learn to identify these cues and be able to associate them with infested cells, and then to investigate. It's very hard to see often. So these are 2 colonies. One's got an infestation rate of 4%, the other one, the recapping rate of 52%. It's surviving, and this one is susceptible. And from the outside, it is often typically very difficult to tell the difference. And this is a bit of a pain in our arse for scientists and beekeepers, because to identify often these cells, you have to open them up. This is what we do a lot. Hopefully we'll do it so you don't have to. But these are 3 cells from the outside, but when they're opened, the top cell is normal, like Melissa showed yesterday, with a nice silky cocoon. And then these 2 different sizes have been recapped, one with a fairly small hole in the middle, and then one with a much larger hole in the middle. So they vary in size. And they're fairly easy to detect either with a magnifying glass, but you've got to be careful. You can't tear these cells, and it takes a little bit of practice to open these. And we run recapping workshops, and yeah, it is not easy to learn well, it is easy to learn to do this, but you need patience. Sometimes you get a colony, like Melissa said, that you can actually, you've got a pretty good idea. And this is a good example. You can see where the cells have actually, the wax has completely changed colour, and these will almost certainly be recapped cells. So what beekeepers, some beekeepers do, they don't look for, well, they look for evidence of recapping, but it's very difficult to see, so they look for evidence of cells being open or half-eaten. So the size of the recapped hole actually differs. You tend to get big holes where the cells are infested and little holes where the cells are not infested. And I'll explain why that is in a minute. So bees can detect infested cells, what evidence there is. So this, as soon as I saw Melissa's paper in review, I thought I'd very quickly fill in the gaps and then offer them. So the first thing that we had a look at is what happens in varroa naive populations. So populations have never had varroa. So we looked at Australia, Colonsay, and the Isle of Man. These populations, each one of these bars is a percentage of recapped worker cells, and it's of many colonies. And the average is a few percent overall. Now, let's have a look at susceptible ones. Now it gets a little bit more complicated. So we have Melissa's data, original data from Norway, Sweden, and 2 populations from France. And we've added in our UK data that one of my students, George, did. Now we have the yellow bars on non-infested cells, and the red bars are infested cells. And you can see that each of these 5 populations can all detect infested cells. Because infested cells, there's more of them being detected than non-infested cells, and there's a lot less infested cells in the colony. So that's really interesting that they already have this ability to do this. But let's have a look what happens when you compare it to resistant colonies. Now, you can really see that these colonies are getting good. So this is pretty much, I think, all the data we have at the moment. So we've got Melissa's 4 original colonies, populations from Sweden, France, Norway, Sweden. Sweden, France, Norway, France. Then we have England, some work we did here. We have Cuba, which has got European bees. So these are all European bees up to here. And then in this last batch, we've got Africanized and African bees, because just people treat these differently. So these are Africanized bees, and then 2 different subspecies of African bees. And you can see that particularly the Africanized bees, they've been doing this a long time, and they are good at it, really good at it. I mean, you know, we're hitting levels that recapping on averages in a population can be sort of 60, 70, 80% almost. So they are really going at this. But also, you will see that the non-infested cells, they are also increasing. And that is a strange thing. And I'll discuss that just now. The other thing is drone cells. So we just done 2 sets of drone cells at the moment. And I can tell you there's no significant difference. It looks like it, but I didn't put the error bars in. But they are not recapping drone cells. They're not actually targeting the mites for some reason. So I got my poor student to solve the problem of the open cells. So why are non-infested cells recapped? And this is the answer. They're clustered. Okay? So they're clustered, their recapping is clustered around infested cells. And what we think is happening is that the odours are very, very difficult to detect, because in a beehive, lots going on. You know, the air currents are moving, all the bees are moving across the surface constantly. So anything that's coming from a certain point will be distributed very quickly. And so they can smell in the area there's a problem, and they start opening cells to try and pinpoint the right one. Where there's no infested cells, there's no odor, so there's no point to do any recapping, which is what we find. So that is why you get a lot of non-infested cells opened, and that's why you need recappers to make sure that these are not removed. So the framework that we have built is you get increased mite detection, and that causes two things: increased recapping and increased brood removal, which I've not talked about, but this is very important. So increased brood removal or cannibalization, that reduces the amount of mite reproduction, that reduces the viral and mite burden on your colony, and increases colony survival. So where's the evidence for this? So this is a study that my student did. She looked at every 60-odd studies that had already been done. She looked at all the data on the colony level, and this was completely independent. We didn't have any data in here. So recapping, the average is 33% in susceptible colonies. It's higher in resistant colonies. So that's great. Also removal in susceptible colonies is about half of what you get in resistant colonies. Now, when you get removal, you lose brood, chance to produce brood. We know that each mite can produce about 25 eggs, reproduces 2 to 3 times in its lifetime. So this very, very simple chart tells us everything. All this does, it says that if there's no brood removal, 0 infested cells removed, the mites can produce 1.4 offspring maximum. And that was from all the work we did in the preceding years. If there is 100% removal of bees, of infested cells, there'll be no offspring produced. And if there's about sort of 50%, you get about 0.65 a reproductive mites produced per cycle. So we were able to do this, and this then explains, I mean, you just take my word, I suggest you read this paper. It is written fairly well in English, that the increase in fertility rates we've always found in resistant populations. So in resistant populations of 45% of the mites are infertile, and in susceptible colonies it's a lot less. And the reason for that is that they are being removed and they are losing their offspring. So we saw yesterday that these numbers, this average number of the number of offspring that they produce per reproductive cycle. If you just think as 1, above 1, and your mite population increases, below 1, and it tends to just be stable. So the work that I did way back, 30 years ago, in the UK, we got a value of about 1.3. It's never really 1.4 because there's a little bit of mortality as well, male death. But then this is Melissa's data, it's over 1. But all the resistant populations, so this is Melissa's data in Europe, Cuba, Brazil, Mexico, they're all below 1. So there's high infertility. They're not reproducing very well, and they're not reproducing very well because they're being removed. The infested cells have been detected and removed. So this is some better evidence from Cuba and Hawaii. So we found that in Cuba, 80% of cells that we infest artificially are being removed, detected and removed. The control cells were quite high. we don't know why this is, but we do know that we don't know when we put a mite in the cell if there already is one in the cell. And it was at the period of very high mite loads in these particular colonies. A lot is very low. And it was also the first time this student did this, so maybe in some errors there. When we repeated this in Hawaii, we got about 70%, did it twice, 70% removal of infested cells, but the controls were a lot lower. So they are very good at this. This is the evidence that the virus load drops. So we have here, it's a log scale, so it doubles. So it's a 10,000, 100,000, goes up very fast. But these values here, about 10 to the 8, are pretty much what you have in your colonies that are healthy. The bees are healthy. They're quite happy with this amount of virus. And so in Apis scutellata, capiensis, and Africanized bees, the level of virus is much lower than in treated populations of Apis mellifera and in the USA and the UK here. And it's very easy to see why, because if you're removing the bees that have the varroa, that have the virus, then you're stopping the transmission cycle. So you're just stopping the offspring, passing the virus on to other ones. Yes, some of the bees will ingest the virus when they're eating it, but this is a natural transmission route for bees, and they're very good, have very good defense systems when they swallow virus rather than have it injected via a mite, because injection is not good for them. And one thing that we found that was accidental and very exciting was in 1996, 1999, when we were originally doing the work in Mexico and Brazil on resistant colonies, the average infestation rate of worker brood was 20%. Now, 20 years later, that had dropped to 4%. Cuba was in the middle of that, it's about 13%, it's not been going that long. We think, although there is very little evidence for this at the moment, that the mite populations are starting to avoid going into worker cells of resistant colonies because they're getting removed, just like what happens in Apis cerana. And that would be amazing if that was actually true. But eventually these resistant colonies are getting down to very low infestation rates, so very little damage is being done in the colony. And hence you just don't need to treat these. I'm going to very quickly finish just as beekeepers. We were very lucky to go to Cuba. Nobody knows anything about Cuba. This is a forest in Cuba. The vast majority of Cuba are covered in forests. And this is a very unique forest for you Danes and Scandinavians, because this is a palm forest. It's a paradise forest. And it's a paradise forest not for you, but for honeybees, because they produce these enormous florets, these flowers. Well, the sort of pod, it's the flower, it's not a metre long, but the actual structure's over a metre long, and it has got thousands and thousands of pollen and nectar producing flowers, which the bees go crazy for. And so in Cuba, they have pretty much unlimited forage for their bees. They have a lot of colonies. This figure's wrong, I said. It's now 221,000 colonies in Cuba. This is one of the queen rearing apiaries we went to. Very professional. Queens there cost €3. It's a very poor country, remember. And they have European honeybees. They are not Africanized. They've been isolated for 60 years. They have European honeybees. It's been tested many times. They have the varroa mite, the same varroa mite as we do. It's the Korean type. And they have deformed wing virus. Why were we there? 20 years now, no treatment for varroa. Average 70 kilos of honey. That's average across the whole island, and it's up to 100 kilos in good conditions. The Cuban government decided centrally when varroa arrived not to treat. Yes, sure, they lost 40,000 colonies over a period of about 5 years as the varroa spread and killed colonies and resistance arose. But they had no other choice. And now they're in this situation. They were allowed to do drone trapping to try and save their colonies, which helped. And now they're in this situation where they don't treat, they don't have to treat, and they have extremely healthy and extremely calm and extremely productive colonies, which is what we all want. This is the future now that's happening in Cuba. And their populations are growing by about 10 to 50,000 colonies a year now. They were down to about 160,000 at their lowest point, and it's still increasing. Just to finish on, we were recently doing a queen swap experiment, taking varroa resistant queens and putting them in varroa naive hives and seeing if we could do that. So whether it was the workers that were important or the queen that was important for this trait. The evidence, we're just starting to write it up, but basically if you have a varroa naive queen put into a colony with resistant workers, after 1 year, the number of mites on the bees is very high. But if you put a resistant queen in varroa naive workers, they become a resistant colony very quick, which is fantastic. So it looks like the trait is hardwired in, eventually becomes hardwired into the queen. What can you do? How can you do this? Well, we'll talk more about in the workshop later today, but basically, I mean, there's loads of methods, but with just easy one. Half your treatment, reduce its effectiveness. If you're doing oxaliates, go into a biotechnical control. Double your monitoring. You've got to start really looking at your monitoring to avoid losses. This is one very quick example because I'm running out of time. So this is Steve Wiley's Westmorland Beekeepers Selection Program. What they did, they stopped treating. They all decided to stop treating in this association. And then what they started to do was to monitor things. They're very, very big monitors, this group. And they can't do recapping or anything, so they were monitoring what was on their floors. And these are evidence of pieces of pupae that have been removed. And they call them hockey sticks, which are the pieces of antennae that come out, and they just look like a hockey stick. They also count the number of mites on the floor. And then they look at how the mites are building up. And so they've got a few hives here. So the hive 4, 6, and 1. It's not building up very, very highly. So they breed from these. But hive 8 is really starting to increase. Get rid of it. And they've used this philosophy for 5 years now. And this is a typical resistant colony that's been studied for an entire year. They're 5 years in now, and this gives you an idea. Evidence where they've seen uncapping, where they've seen sort of evidence of pupil removal, the mite flow. These guys are a bit obsessed with their sort of observations, but this is great. And now they're 5 years in, and they're pretty much without treatment. And other groups are starting to join them. And they just made the decision to change. Independently, I've met these groups recently. So the do's and don'ts, I suggest use local bees. Don't move bees, it's not good. And don't stop treating. Okay, that's not the answer. You stop treating, you'll lose your bees. You've got to do this gradually. Or if you do stop treating, you have to come up with very, very high and strict monitoring systems. We've written practical guides of how you can measure recapping, really, if you want to. There's a YouTube thing, but this is really, you know, you've got to have plenty of spare time. You'll be retired and really want to do this as a group. I'll measure insertions. There is a lot of information now online. We are in April, we are going to release a website, scientific website called varroaresistant.uk. Don't look now, it's not there. At our big conference, and it will have all the scientific information and advice and examples which will develop it, and you're free to look at it. I will let Fleming know when it goes live. And I'd like to thank everybody, particularly the beekeepers and all the people. There's a lot of work involved in here. I'm not the one that does all the work. I'm just not physically possible. This is my team over the years that have been doing various things. They've all gone on. They're all funded by beekeepers, and they're all gone on to work either in bees or on COVID or looking at mosquito viruses, but you know, doing things which benefit people. So that's great. So thank you very much for everything, and I'll call it a thing. Stop there.